So after the plant makes organic molecules, it has to transport these to the different parts of the plant. This is a process known as translocation. In flowering plants, the tissue that transports these molecules and these ions is called the phloem. It is made of several sieve tube elements that are long and thin structures arranged from end to end. The walls are perforated to form sieve plates in order to filter the material that passes through them. Associated with them, we have companion cells, and this is the structure of this system. So plants produce sugars during photosynthesis and then transport them from the sites of production, which are the leaves. They're known as sources. They're transported from these sources to the places where they will be needed for use or for storage, known as sinks. So the transport is from sources to sinks. Sinks can be anywhere in the plant, so the translocation of molecules in the phloem can go in any direction, really. The transport includes molecules like sucrose and amino acids, and also inorganic ions like potassium, chloride, phosphate, and magnesium. We are still not sure of the exact mechanism by which this translocation is achieved. However, right now we are most convinced with the mass flow theory, which can be divided into 3 phases. The first phase is the transfer of sucrose into the sieve elements from the photosynthesizing tissue, like the leaf. So in the cells that have chloroplasts, we make sucrose, we as plants, I mean. The sucrose is going to diffuse down a concentration gradient through facilitated diffusion into companion cells. In these companion cells, we have hydrogen ions being transported by active transport into the spaces within cell walls, and these hydrogen ions will diffuse down a concentration gradient from these spaces within the cell walls through carrier proteins into the sieve tube elements. Along with them, we have the transport of sucrose molecules in the process of co-transport that we talked about previously. We have protein carriers here that help with this. They are known as co-transport proteins. So this is the first phase by which we transfer sucrose into the sieve elements from the place they were synthesized.